All right, we're going to talk about photosynthesis today. Little mini lesson in photosynthesis. I'm going to reduce that and make sure I'm still recording here. Are we still recording? Okay. Photosynthesis is an overview slide. First of all, it's a remarkable ability of an organism to harness light energy and use it to drive the synthesis of organic compounds. Okay, so this occurs in plants, of course, but we also know that photosynthesis can occur in other autotrophic organisms, such as some bacteria, some protists, and definitely algae, which is included in that group. So plants, we call them producers or autotrophs, also known as photoautotrophs. And the process of photosynthesis is an endergonic reaction. Okay, so when you see the equation written here, it is endergonic because the electrons that are in this process increase in potential energy as they move from water to sugar. Okay, so the redox reaction looks something like this. Okay, so carbon dioxide plus water in the presence of sunlight is going to produce glucose and oxygen. So because it's a redox reaction, the carbon dioxide is going to become reduced, okay, as electrons are added to that to form our sugar. The oxidation, okay, is because water, due to the splitting of this, is going to become oxidized and form our oxygen at the other end. So the main thing that's going to happen is we're going to have the splitting of water. That's going to play a huge part in our photosynthesis reaction. So water will be split, electrons will be transferred from that water, along with some hydrogen ions, known as protons, to the carbon dioxide, reducing it to our sugar. So we're trying to form that. Okay, I have no idea why it's doing that. But, anyway, so that's our overview. This is going to happen in the chloroplast. Remember that plants definitely contain membrane-bound organelles. So if you were comparing this to, let's say, photosynthesis in a bacteria, they wouldn't have the chloroplast because they lack membrane-bound organelles, but it would take place in their cell membrane. They possess chlorophyll molecules in their cell membrane, which kind of acts like a pseudo-thylakoid. So the chloroplast structure, a little bit about it is, you can see that here's one chloroplast, and inside of it, let me see if I get my little tools here. Get my little tools. Sweet. We've got these little bitty stacks like this. I call them little poker chips, but each one of those is known as a thylakoid. And so a chloroplast might have many thylakoids stacked up into one giant stack like this. The whole stack is known as a grana. Okay, or granum for singular, grana for plural. And so each chloroplast would have several stacks of these grana within them. Okay, now you're going to hear me also talk about the word stroma, because that's where one part of photosynthesis takes place, known as the Calvin cycle, and it's in this space out here between the stacks of grana, or outside of the thylakoid. Now one thing that's really cool is the inside of the thylakoid. On the inside, there's a special little concoction of chemicals inside of there, so it's called the thylakoid space on the inside, which is unique on the inside compared to the stroma on the outside. So we're dealing with the chloroplast, and there are many chloroplasts inside green plants. You will find these not only in the leaves, that's where the highest concentration would be, but also green stems, as well as in fruits that have not ripened yet. You would find chloroplasts in those as well. Okay, so photosynthesis, we just said it's in a chloroplast, but just to give you a little picture of the leaf anatomy, so you know where this is taking place. Here's a little picture of a leaf, and this middle section right in here is called mesophyll. Let's see if I can write using this pen. Okay, meso, P-H-Y-L-L, which is that middle portion of the leaf, and you can see all the green chloroplasts located in here. So these cells have lots of them. So this is the area where photosynthesis takes place. Okay, now you see some of these down here are loose cells. Okay, so you have kind of a lot of airspace in here, because near the bottom of the leaf, the bottom surface, this is where our gas exchange is going to occur. So you can see with this arrow showing here, the little blue cell would indicate something called a stomata. The stomata is the opening on the underside of the leaf. This is where CO2 would come in, okay, to the stomata, come into the airspace and be used in those cells for photosynthesis, and then oxygen, which is our product, comes out of that same opening. So that stomata is used for gas exchange within a plant. Okay, now remember they also need water. That's another reactant for this process. So water would come up through the root system, be absorbed, and eventually come out the vein, because in the vein you've got xylem and phloem, and the xylem is the part that transports water and minerals. So you'll be able to get your reactants for photosynthesis through that part of the leaf to enter for this process. Okay? All right. Let's see if I can get this up here. Okay. Let's see. Get rid of this. All right, next slide. This is a great picture. I thought this was a really good one. I think you have this in your book, but it's a great overview of the process of photosynthesis. So let me see if I can minimize that. Bring back up my pen. Sorry for changing back and forth, but I'm constantly writing on this. Okay, so what you see in this picture, here's the chloroplast. Here's the thylakoid, okay, or that's a sac of granite. Here's the stroma over here. Okay, so we've got two things taking place. Okay, we've got actually two parts of photosynthesis. So photosynthesis is not a single process, but two processes, each with multiple steps. The two stages are known as, okay, number one, right here, called light reactions. The photo part of photosynthesis is actually because of the light. And then we've got Calvin cycle over here, which is where we get the synthesis part of the word. So photo, light, synthesis is Calvin cycle because this is where we're synthesizing our sugar. So the light reactions over here, just looking at this part, is where solar energy, okay, so here's your big sun. Don't laugh at my drawing. Okay, so solar energy is going to be converted into chemical energy. Okay, so in order to do this, we have to start with water. Here's one of our reactants. So water in this step of the light reactions is actually going to be split. Okay, so it's the main splitting of water that will provide us with a source of electrons as well as some protons, okay, which are hydrogen ions. Once those are split, we will have down here at the bottom oxygen given off as a product. Okay, so the splitting of water directly is where we get this oxygen source. The electrons and hydrogen atoms are going to be really important because they will be received by this electron carrier called NADP+. It's kind of a cousin to the one that's used in cell respiration known as NAD+. This one has the P, it's got an extra phosphate group on it. I always remember the P for photosynthesis. The NADP is the electron carrier for this process. Okay, so light is absorbed by chlorophyll over here in the thylakoid. So you've got chlorophyll molecules in here, and we'll go through this in more detail in a little bit. But once the light is absorbed, it drives a transfer of electrons and hydrogen ions from water to this NADP+ molecule. They're going to be temporarily stored in that as NADPH. That is one of the main driving forces that will run step 2 over here, known as the Calvin cycle. So that's the main source of energy drive. Okay, the light reactions, in addition to that, will create a little bit of ATP. Okay, so the generation of ATP happens during a process called chemiosmosis, where we're going to have the addition of a phosphate group to an ADP. Remember, we talked about that being phosphorylation, but because it's in photosynthesis, this is photophosphorylation by adding that P on an ADP to create ATP. Okay, so thus light energy is initially converted to chemical energy in the form of two compounds. We end up with NADPH, and we end up with ATP. That's our main product of the light reactions, and those two things will go and run and power the Calvin cycle. Now you'll notice once the Calvin cycle occurs, what we end up with over here is we need our sugar. Okay, so the general formula for our sugar is CH2O. And all intents and purposes, it's really going to be glucose. The reality is it's a 3-carbon compound that can be converted into glucose, but to make this simple, we're just going to talk about the production of glucose. So after this happens, the Calvin cycle, you end up getting recyclable products over here called ADP and an inorganic phosphate, which can go back to the light reactions and be used again to generate more ATP. And then we end up with the NADP+ that can then recycle back over to the thylakoid and be used once again when more light comes in and more water comes in as reactant, and we can split those and the whole cycle starts over again. So you can see the recycling of those products in the middle here that occurs between the light reactions and the Calvin cycle. Okay. Now I want you to notice I wrote the words oxidation reduction above, because remember this is a redox process. So the oxidation part is the light reactions, whereas the reduction part is in the sense of the Calvin cycle, because we are reducing by the formation of our sugar, okay, because our sugar is going to have the electron energy is within that, so that's reduced by receiving electrons. Okay, so just to sum that up before I switch slides, photosynthesis, when we get into more detail a little bit, we'll have two parts. Okay, part one, what is it? Good job, light reactions. Part two, over here, Calvin cycle. By the way, Calvin cycle, I didn't mention this earlier, was discovered by Mr. Melvin Calvin in about the late 1940s. Sometimes this is referred to, and it's kind of falsely referred to this, but sometimes they'll call it the dark reactions. The reason that it's kind of a false sense of the word is when you say dark reactions, most people think, oh, it happens in the dark, when in reality most plants are doing the Calvin cycle during the light time because the light is what must be provided for step one. And really the more time, like the more you do step one, the more times it can be run over and run the Calvin cycle. But Calvin cycle could actually happen in the dark, but it just happens more often in the light because of what plants are doing in that step. Sometimes it's also called the light independent. That's a more politically correct term. Light independent reactions is another word for Calvin cycle because it's independent of a light source. It could happen in the dark, even if you take that light source away. Okay, so let's get rid of this little pen. Let's see if I can get my thing back up. All right. So I added this slide in just so that you would know that sun is our biggest energy source. It's a giant thermonuclear reactor. So energy is coming from fusion reactions similar to those in a hydrogen bond. When light hits matter, it can be reflected, transmitted, or absorbed. So all light coming off the sun, when it hits a plant, it has a choice to be reflected, transmitted, or absorbed. Okay, so in order to understand this, we have to talk about the visible spectrum a little bit. Okay, so the light reactions, like we said, happen in the thylakoid membrane. So when the sun comes down, okay, and by the way, visible light is a very small section of this visible spectrum. You can see that we've got everything from gamma rays, X-rays, all the way to short waves and radio waves are down here on the end. And so you've got like a huge part of where light falls, but the visible light is right in this small section. And so you see these are nanometers that are measured, wavelength. And let me move this little thing so I can get this up here. Visible light is a small portion, like I said, of the electromagnetic spectrum. Light is absorbed by chlorophyll, but that's not the only pigment found in plants. There are other pigments called xanthophylls, carotenoids, you've got chlorophyll A, chlorophyll B. So pigment molecules are present in the plant, and it will power the reactions. Okay, now light that is not utilized by the plant is reflected. So plants that appear green are green because they're reflecting green light. Okay, they're actually absorbing the parts that you don't see. And the part of the spectrum that plants really, really like, their photosynthetic ability increases, is over here in like the blue-violet area and then in the red area, red part of the spectrum. So light energy can be measured in terms of photons, which each have a fixed quantity of energy that's inversely related to their wavelength. Okay, so the longer the wavelength, it's not the more the photon energy, it's actually inversely related to that. Okay, so we've done our chromatography lab, so I kind of threw this up here just to give you an idea that when you look at pigments, we did this in class the other day, where you can extract pigments using chromatography solvent, and you can see that several things are going to contribute to the moving up of the pigments on the paper. One we talked about was the hydrogen bonding ability, and then others is how polar or nonpolar they are and whether they can dissolve in a solvent. So pigments are substances that absorb visible light. So we've got chlorophyll A, chlorophyll B, we've got carotenoids, and in this sample we have xanthophyll as well. Chlorophyll A is the one that mainly absorbs blue, violet, and red light, and chlorophyll B absorbs blue and orange light. Carotenoids are other accessory pigments that can also be used in photosynthesis, but you can see like in this part when we did this in class, your chlorophyll B has the bottom band. It doesn't pull quite as high up on the paper. Chlorophyll A goes this high, and then xanthophyll, and then carotenoids. So go back and read your lab sheet, and you can see why some of these travel farther than others. I don't want to answer your lab for you, but make sure you go back and look at that. Okay, so to be able to measure wavelengths and how much light plants are capable of either absorbing or transmitting, we use a tool called a spectrophotometer. Now, we're actually using in our class calorimeters, not spectrophotometers. SPEC measures light transmittance, whereas the colorimeter that we're using is measuring absorbance. So, it's just a way for you to measure different wavelengths of light. Okay, let me get my... Okay, so, see if I can get this. Okay, so now we're going to look at a little experiment. First of all, you see here the little graphs are labeled absorption spectrum, action spectrum, and then this guy named Engelmann, his experiment. Okay, so first of all, a graph plotting a pigment's light absorption versus wavelength is called an absorption spectrum. So the absorption spectrum of chloroplast pigments provides clues to the relative effectiveness of different wavelengths for driving photosynthesis, since light can perform work in chloroplast only if absorbed. So what you see up here at the top, it shows the absorption spectrum of chlorophyll A. Okay, you can see chlorophyll B, and you can see carotenoids. Okay, so the spectrum of chlorophyll A, which is the one that I'm particularly looking at, this one right here, suggests that violet blue and red light work best for photosynthesis. So look at how this spikes up here at violet blue, and then over here at the red. Okay, so you can also see that green is least effective. Okay, so the absorption rate is decreased around the green area. So that gives us some clues as to which part of the visible spectrum that the plants are most concerned with and using. Okay, this can be confirmed by an action spectrum, okay, this bottom one, for photosynthesis, which profiles the relative effectiveness of different wavelengths of radiation in driving the process. So it's prepared by illuminating chloroplasts with light of different colors and then plotting the wavelengths against some measure of photosynthetic rate, such as carbon dioxide consumption or oxygen release. So the action spectrum for photosynthesis, this was first demonstrated by a German botanist called Theodor Engelmann. This happened in 1883, and before equipment for measuring oxygen levels had been invented, he performed a clever little experiment where he used bacteria to measure the rates of photosynthesis in filamentous algae. Okay, so he used little filamentous algae. So the way he did it is kind of cool. Is he used these little filamentous algae and had light that passed through a prism. He exposed different segments of the algae to different wavelengths. The bacteria he was using were aerobic bacteria, which actually concentrated near the oxygen source. Okay, so to determine which segments of algae were releasing the most oxygen, which then told him what was photosynthesizing the most. So ironically, when you look at this, bacteria congregated in the greatest numbers around the bluish purple, violet, and then over here around the red. So that led us to also then support this original absorption spectrum up here, which says that photosynthetic organisms prefer the chlorophyll A specifically, prefer the wavelengths over on the violet, blue, and the red area part of the visible spectrum. So it was a cool little experiment that he did to illustrate that. Okay, so now we're going to get into some specifics. We're going to take and break down the light reactions, and then we're going to break it down into the Calvin cycle. So we're going to break up the two steps. So this is a really cool diagram. If I can move this down. The first thing that you see is we're going to start off with the light reactions with a photon of light that's coming in. Okay, so here's your light energy. As the photon comes in, by the way, there are two photosystems that we're going to deal with. I forgot to mention that. Okay, there's two photosystems, and they're kind of backwards because you would think you would study photosystem 1 before you study photosystem 2, but it was actually they were named in the order in which they were discovered. So photosystem 1 was discovered first, but it happens later. So photosystem 2 is what's happening here in this picture. This is the first step of the light reaction, which starts with our light energy coming in. Okay, so as this comes in, energy will be passed from one pigment molecule to the next, to the next, to the next, to the next. You can see this is passing on of energy, photon energy, to different pigment molecules. It finally will come to a special pair of chlorophyll A molecules located in what we call the reaction center. Okay, so let me see if I can get my pen up and running. Oh, maybe if they'll let me do that, and then get my pen. Here we go. Sorry about that. Okay, so this is photosystem 2. And what we have here, let me just kind of give you the breakdown of this, is we've got light harvesting complexes, which are these big purple blobs that you see here. And we've got a reaction center complex, which is this thing in the center. Now, embedded with that, we've got all these green, which these are chlorophyll molecules. They're different pigment molecules along. Right here in the middle of the reaction center is our special pair of chlorophyll A molecules. Those are really, really important. Okay, so this all starts with the photon. This has to be the sunlight coming in. Okay, so photon energy comes in. Energy is passed from pigment molecule to pigment molecule all the way along, which will then eventually be received by our special chlorophyll A molecules. Now, these actually in photosystem 2, the preferable one, are called P680s. Those are the ones that's the part of the visible spectrum that they prefer. The electron in each is excited to a higher energy state until it's transferred to a primary electron acceptor. So the primary electron acceptor is this little box. Oops, I did not mean to do that. This little box right here. And so that's going to accept electrons as these are raised to a high state. So once again, photon energy starts the energy coming down. This excites, energy is passed to different pigment molecules, gets spread or passed to chlorophyll A, who then raises that electron to an excited state up to our primary electron acceptor. Okay, now, in this same process, we are also going to split some water. Okay, so let me see if I can get all this mess off the screen. There we go. Okay, so to replace the electrons that P680 passes up, we're also going to use our water right here. So water split will then replace the electron lost by P680 because the electron goes up. So we're going to have light providing some energy source. We have the splitting of water providing another energy source, and that's also where we get some hydrogen ions. Okay? All right. Okay, so one thing I wanted to also mention here is that if this photosystem was if this was photosystem 2, the special pair of chlorophyll A molecules, I mentioned this over here, are the P680 because that's the nanometers that they prefer to absorb, which is the red part of the visible spectrum. But when we talk about photosystem 1, it's actually a special pair of P700 because they prefer a far red part of the visible spectrum. So they're essentially very similar to each other. They're just slightly different on the spectrum, but they're both going to have that special pair of chlorophyll A molecules. Okay, something else to consider, just because I want to give you a little analogy. I know sometimes it's hard to understand that when this light energy comes in, that the energy is passed from pigment molecule to pigment molecule, but if you think of it as like a wave in a sports stadium, like if you're at the Texans game, and everybody starts doing the wave around the stadium, it's like one person does it, then goes to the next, to the next, to the next, to the next, or the roller coaster that we do at school. That's kind of the same thing. So energy is being transferred, drop, and then eventually it's passed to that special pair of chlorophyll A molecules. And that's in the reaction center complex. Okay, so passes it up, goes to that primary electron acceptor. I also wanted to mention here at this primary electron acceptor, that's our redox reaction because that electron acceptor is accepting the electrons. So that's being reduced by receiving that electron. Okay? All right, let's look a little bit more closely. So we see this much better picture where we can see both photosystems interacting together. Okay, so down here we've got photosystem 2, and then we have photosystem 1. In between both of them, you'll see there's the electron transport chain between them. Okay, now I don't know if you noticed in the background, see this grayish membrane? Those are phospholipids, just like we talked about the phospholipid bilayer with the cell membrane. This is the thylakoid membrane, which is this phospholipid bilayer. That's going to play an important role in building up a gradient with these hydrogen ions down here on one side of the membrane. Okay, so let's talk about how the two photosystems work together. Okay, so in this diagram, we've got photosystem 2, photosystem 1, and then over here off on the side, you can see this is where the ATP is going to be generated. Okay, remember, big picture again, what we're trying to do in these light reactions is we are trying to create NADPH, which you can see is right here, and we're trying to create ATP. Those are the two products that we want to gain from the light reactions because those need to go over to the Calvin cycle for part 2 and drive the Calvin cycle, okay? So the way this is going to happen, and this kind of looks crazy, and we're going to take it in several parts, but photosystem 2 occurs first over here, okay? So here's our reaction center and everything going on here, and then here's photosystem 1. Okay, so photon of light is number 1. Okay, so here's the light coming in. It strikes the pigment molecule in the light harvesting complexes of PS2. I'm going to call it PS2, which sounds like the gaming system, but it's easier for me to say PS2. So the photon comes down, strikes the harvesting molecules, boosting the electrons to a higher energy level. Okay, so it's passed on like a wave. Okay, boosting the energy level. As it falls back down to its ground state, the electron in the nearby pigment molecule is stimulated, raising it to an excited state. This process continues with the energy being relayed to other pigment molecules until finally it's reaching the P680, which is right here, the special pair of chlorophyll A molecules. Okay, once it reaches that, we just said a minute ago, it's going to be excited, raised up. Okay, you can kind of see where my arrow is right here. It's raised up. Goes to that PS2 reaction center complex. Okay, and it receives that electron as that electron acceptor. Okay, that's the primary electron acceptor. We can refer to the resulting P680 right here, because it just passed on its electron to this acceptor, we now call this one P680 plus. Okay, it's plus now because it gave away its electron. Okay, so at the same time, what's going to happen next is we've got water. This is one of the reactants of the light reactions. So an enzyme, because we have lots of enzymes that help out with this, is going to catalyze the splitting of water. When we split water, we're going to get 2 electrons. Okay, if I can get my little Moby up. This is frustrating. Let me see. Hang on. All right, hang on. Let me see if I'm going to pause for a minute so I can change pen colors. Okay, we're ready. Sorry about that. Okay, so down here we've got the splitting of water. When we split water, we will get 2 electrons. Okay, so we've got like E negative. We've got 2 of these. We've got 2 hydrogens, okay, right over here, and we have an oxygen atom that are formed. The electrons are supplied by one of the P680 plus pairs, each electron replacing one transferred to the primary electron acceptor. Okay, so each one of those electrons over here can go back and fill in to the one that P680 already gave up here to this receiver. Okay, so that electron will keep supplying that energy source. Okay, by the way, P680 plus is the strongest biological oxidizing agent. Its electron hole that it leaves when it gives up its electron, it must be filled. This greatly facilitates the transfer of electrons from the split water molecules. The hydrogens are released into the thylakoid lumen. Okay, do you see all these hydrogens down here creating this big gradient? So the hydrogens are released down here to what we call the lumen. It's one side of the thylakoid membrane. The oxygen immediately combines with an oxygen from another splitting water, because you're going to have constant splitting, because plants use a lot of water. So we'll get more oxygens by that, and those can eventually form O2, which can be released, like we said, out of the stomata of the leaves of the plant. Okay, now each photoexcited electron passes from the primary electron acceptor, so from this, of PS2 to PS1. Here's PS1 over here. Okay, so this is PS1. Here's PS2. So we have to get that electron to go from here over to PS1. So to get there, it's got to travel across this middle section that you see, which is an electron transport chain. Okay, right in here. So the components of this are actually very similar to what we are going to look at when we study cell respiration, which occurs along the cristae of the mitochondria. So this electron transport chain along here is made up of a couple little, what we call electron carriers. This one's called PQ. Okay, not that you have to memorize like the name of this, but it's plastoquinone. And then there's also another one, a special kind of protein called PC over here. Okay, so PQ and PC. The exergonic, look in the word, the exergonic fall of electrons that is occurring across these, okay, to a lower energy level, provides the energy needed for the synthesis of ATP. Okay, so the energy from those falling is going to build up this hydrogen gradient over here and provide a little bit of energy for us to synthesize our ATP over here at the end. So as electrons pass through we call this the cytochrome complex, which is this thing right here. This is the cytochrome, that purple thing in the middle. So we've got a PQ, a PC, which are both electron carriers and little proteins, but then right in the middle, this fat one, we have something called cytochrome. Okay, cyto, and then it's chrome. So as electrons pass through that complex, hydrogen, see this hydrogen right here, is pumped through into the thylakoid lumen, which continues to build up a big gradient of protons. Okay, that buildup of that gradient is what leads to a process down here at the end called chemiosmosis. Chemiosmosis is the transfer of those hydrogens through a special little protein right here called ATP synthase. It goes right through that to the other side. Well, as it does that, that's the energy gradient that's needed to get a P to stick to an ADP to create an ATP. So that's where our ATP source comes from. Okay? So meanwhile, there's a lot of stuff going on. Meanwhile, light energy has also come in here. So light energy is continuing to come in to PS1, into that reaction center, which then simultaneously starts to excite the electrons, which are going to excite the P700 in this area. Okay? So those chlorophyll A molecules that are located there also get excited, which are then passed on to PS1's primary electron acceptor, which is right there. Okay, that then creates a hole right here in the P700 spot, which we now can call P700 positive. Okay, so now that can act as an electron acceptor, accepting an electron that reaches the bottom from PS2. Okay, so this electron coming through here can now be received by P700, because it already passed its electron up to its acceptor up here. Okay, so there's a lot of stuff going on. So we got the light energy coming in, passing on more molecules down here in PS1. Okay, those can be passed on to the P700 right here, which can be raised up to their excited state to go to their electron acceptor. And that hole is filled by the electrons coming over here across the electron transport chain from PS2. Okay, so once that happens, now we're ready. Over here at the end, we're about to finish this, y'all. Over here at the end, at PS1, there is an enzyme that exists over here. Okay, there's an enzyme. The enzyme's name is called NADP+, which sounds like this, reductase. Okay, reductase. It's a special enzyme over here. And that NADP+ reductase catalyzes the transfer of electrons from a special little dude here. It's another protein called SD. So the enzyme NADP+ reductase catalyzes the transfer of electrons coming across this SD molecule, okay, to NADP+. 2 electrons are required for its reduction to form NADPH, and that's what these blue things are illustrating, is those are the 2 electrons that are needed to reduce that. Do you know what's so amazing is that NADPH molecule, no joke, is actually at a higher energy level than water itself, and water is pretty energetic. Its electrons are more readily available for the reactions of the Calvin cycle than those of in water. Okay, so this process is also removing some hydrogens from the stroma side. Okay, so if you imagine this, we take some of these hydrogens over here, that even leads to more of that concentration gradient where we have a lot of hydrogens on this side, the lumen, and a less concentration of hydrogens over here on the stroma, which that still goes back to lead to our buildup of our formation of ATP by pumping these H's across that gradient. Okay, so that's a lot of stuff happening on there. Those are the light reactions. So the next slide should be pretty easy. Let me see if I can get out of this little mode with my pen. All right, so this picture, we've already gone over it, basically, just so you know. This is just kind of taking what we did and drawing it out to another diagram. So you can still see, same exact thing we talked about. There's Photosystem II, Photosystem I. So everything's happening. If you need to review it, go back and look at the other one. I don't want to keep going over this, but Photosystem II has, you can see everything occurring, like bringing the light in, splitting the water. So the 2 things that I want you to understand that we're gaining from these light reactions are we are gaining NADPH and we are gaining ATP so that we can provide a driving force to run the Calvin cycle. All right. Another picture, you can see this is just a bigger diagram of the thylakoid membrane. So you can see Photosystem II here. There's your photosystem 1. This is a good diagram because you can see the cytochrome complex right here. Okay? So going back, if you wanted to just review it real quick, it's the light energy coming in, exciting these little green pigment molecules. Okay, electrons are dropping. They're received by the special pair of chlorophyll A molecules, which raises them up. Oops, I did not mean to do that. Raises them up to their acceptor, electron acceptor, right here, primary electron acceptor. At the same time, we're going to split water. We're going to take some of the electrons from water and fill the void, fill the hole that was left down here at the special pair of chlorophyll A molecules. Okay, the electrons, when they're received by the primary electron acceptor, will be passed along. Okay, so watch my arrow. This is going along the electron transport chain through the special PQ, through the cytochrome complex, through the PC, over here to photosystem 1, received by those special pair of chlorophyll A molecules, okay, this P700, which then raises its electron up to its primary electron acceptor. Okay, so there's a void now, a hole in this one, which this one is now filled by the light that's constantly stimulating over here. Okay, I said that backwards actually. The light is stimulating, those get passed up, the void, the hole is filled by the electrons coming from this. Okay, so pardon what I just said. So the electrons come over from the electron transport chain and fill the hole that was already left after light stimulated these and pass those up to that primary electron acceptor. Okay, at the very end, okay, there you see the electron comes up. Okay, goes to FD. Okay, remember FD. Once the electron gets past FD, we've got our special enzyme. There it is listed there. You can see it, NADP plus reductase. That then helps to take the electrons, join them with some hydrogens to form NADPH. Okay, now this diagram has the ATP synthase in a different location. It's down here, but it can be all along that thylakoid membrane. So remember, the ATP synthase functions because we get the hydrogens that have a gradient that are then going to go out the ATP synthase, and when they go out, it facilitates a P sticking to an ADP, forming a bond, creating ATP. So in this diagram, look at the black arrows. ATP goes to Calvin cycle, NADPH goes to Calvin cycle, and both of those came from these light reactions, okay, of what's going on. Fun, right? Okay, I'm going to come back to this one in a little bit. So we're going to now go through the Calvin cycle of photosynthesis. Okay, like I said, also known as dark reactions or light-independent reactions. This occurs in the stroma. This one's a lot easier. This is the synthesis part of photosynthesis. It's going to use ATP, use NADPH, which were both generated in a light reaction, plus we have to use the carbon dioxide, which remember is a reactant, in order to produce our organic molecules. Okay, so this is an anabolic process. Remember, it's not catabolic, it's anabolic. Also endergonic, because we're going to take that energy and store it in the bonds, our chemical energy of food. There's an enzyme required to do this. It's Rubisco. Okay, not Nabisco, but Rubisco. It's a special little enzyme. And there are going to be 3 steps to the Calvin cycle. Okay, step 1 is called carbon fixation. Step 2 is reduction. Step 3 is regeneration of rubip. Okay, so there's 3 steps to this. All right, don't freak out when you see this diagram. All right, so here we go with the Calvin cycle. Okay, don't freak out when you see this. You guys are you can do this. You're smart. All right, first thing is, okay, let me see. I can get my pen again. Okay, first thing is carbon enters the Calvin cycle. Okay, here's carbon up here at the top. Enters in the form of carbon dioxide and leaves in the form of sugar. So carbon dioxide is coming in, and we're going to have sugars that are leaving. Now, I don't know if you can see, but down here, there's our sugar. Okay, it's going to be down here at the end. But I'm going to move this up so we can see the top part. Okay, so the cycle is going to spend ATP as an energy source. So we're going to have to use some ATP, and we're going to have to consume NADPH as a reducing power for adding high-energy electrons to make the sugar. So we have carbon dioxide coming in, sugar coming out, and we have to use ATP, and we're using NADPH. You can see here's ATP. We're going to use that and NADPH for our reducing power and our energy input in order to come up with a sugar at the end. Now, as I mentioned previously, the carbohydrate that's produced directly from the Calvin cycle is actually not glucose. We always just say glucose because it's easier for students to understand since it's the preferred energy source and it's what plants, typically produce. but in actuality, it's something called, I call it G3P, which is glyceraldehyde 3-phosphate. Okay, so for the net synthesis of one molecule of this G3P, the cycle has to take place 3 times. So if you see up here at the top, it says 3 right there, but they enter one at a time to go around this cycle. Okay, so as we trace the steps, keep in mind that we're following 3 molecules of carbon dioxide through the reaction. Okay, so there's 3 steps. Look over on the side. Here's phase 1, here's phase 2, and phase 3 is over here. Okay, so phase 1 is called carbon fixation. So the Calvin cycle incorporates each carbon dioxide, here it is, it comes in. Okay, it's going to incorporate that one at a time by attaching it to a 5-carbon molecule over here. This is already in the cycle. So this thing's coming around. This carbon dioxide, okay, the carbon is going to attach to this 5-carbon molecule. Okay, the name of this one, I like to call it RUBIP, R-U-B-P. Okay, the real fancy name is ribulose biphosphate, but it's just called RUBIP. So it's already in the cycle. Now, of course, we have to have an enzyme to help out in this. So the enzyme you see listed right here is called Rubisco. That's the enzyme. Go ahead and label that if you have your notes. Okay, that's the enzyme, Rubisco. So once again, the carbon dioxide molecule comes in one at a time, attaches to this 5-carbon compound called RUBIP, and it does it by using the enzyme Rubisco. Okay, they say, according to scientists, that this is the most abundant protein in chloroplast, it's Rubisco, and it's also thought to be the most abundant protein on Earth. Kind of a cool little fact there. Okay, the product of the reaction is actually this thing right here. It's a 6-carbon compound. It's an intermediate form. It's very, very unstable, which is why it says short-lived. They say it's so unstable that it almost immediately splits in half, forming 2 molecules of 3-phosphoglycerate. So we'll get 2 molecules of this because this is so unreactive and it just unstable. I mean, it just basically breaks in half. Okay, the next part, let's do phase 2, is reduction. Okay, phase 2 is reduction over here. So each molecule of the 3-phosphoglycerate that's formed, so we have 2 of these, each one receives an additional phosphate group from the ATP. Okay, so this ATP is going to be broken off. Okay, we are left with an ADP, and we get that inorganic phosphate. So that phosphate attaches, you can see it right here, boom, there it is. Okay, so it attaches to the phosphoglycerate. Once it does that, it becomes 1,3-biphosphoglycerate. Okay, so they name it something different because now it has a bi, and it's 2 phosphates on it. So it's got 2 phosphates on one on either side. Okay, so next, what's going to happen to this, kind of quick right after that, is a pair of electrons that's donated by this NADPH. Remember, there's electrons right there. Okay, so a pair of electrons donated from that, a pair of electrons donated from that reduces this 1,3-biphosphoglycerate, which also loses a phosphate group. Okay, so it's going to donate electrons. It's going to lose one of its phosphate groups. This comes off now. As it loses the phosphate group, it becomes G3P. Specifically, the electrons from the NADPH reduce, if you want to know this, you don't really have to know this, but it reduces a carboxyl group on the 1,3-biphosphoglycerate to the aldehyde group. Basically, it's storing more potential energy. So the potential energy is in this compound. So it's storing more potential energy in that because that's going to be our sugar. Okay, it's kind of essential, actually, that you know that because it's similar to what we're going to talk about in glycolysis. But as a result of this, what comes out is now we have our product. We have our sugar. Okay, so that's the reduction phase, and that reduction phase, remember, comes again from the breaking of the P from ATP. So this is where it's directly used, these products of the White reactions, and we're going to extract some electrons. So we add a phosphate quickly. We then take some electrons. We cleave a phosphate off. So we're reducing this compound into a sugar, and the electrons are actually, the energy is stored within the electrons within that. So then we get our sugar. Okay, for phase 3, over here, regeneration of the carbon dioxide acceptor of RUBIP. This one's actually really quick. In a series of reactions, okay, so some complex reactions occurring here, the carbon skeletons of 5 molecules of G3P are rearranged by the last steps of the Calvin cycle into 3 molecules of RUBIP. So we're going to come back and form some, like 3 molecules of rubit. To accomplish this, though, you'll notice over here what you see on this side, the cycle, spends 3 more molecules of ATP to do that. So the rubit is now ready to receive carbon dioxide again, and the cycle continues. And that, my friends, concludes photosynthesis.